Welcome everyone to the monthly ICG lab series. It's a great pleasure to introduce my colleague and also friend Nils Gehlenborg. Nils is an associate professor in the Department of Biomedical Informatics at Harvard Medical School. He is also a director of the Master in Biomedical Informatics program there. And Nils is originally from Germany, and he studied at the University of Tübingen. And then later he received his PhD from the University of Cambridge and was a postdoctoral fellow at the European Bioinformatics Institute. He then moved to the US to do his postdoc at the Center for Biomedical Informatics at Harvard Medical School, where he still is, and he's associate professor there now. And the goal of his research is really to enable scientists, but also clinicians to interact with biomedical data. And Nils is also a co-founder of DataVision, which is a JKU spin-off company that is located here on our campus. Nils, we're ready for your talk, and we're really looking forward to what you're going to present.
Awesome. Thank you for the introduction, Mark. Thank you also for the invitation to speak to all of you today. I will get started in just one moment. I just wanted to add one slide to my slide deck, which I think might be of interest to you. So let me just pull this up here and get all my windows sorted out. All right. Where are we? So as Mark mentioned, I'm based at a medical school, but I am in an informatics department. So we are really doing very applied computer science research in at least in my lab, and also most of my colleagues are working on computational problems. We have about 15 faculty in our department at this point. It's a fairly new department, it was only launched about 5 years ago. And so I think this is maybe one of the things that sometimes confuse people who are maybe not in sort of computational biology or biomedical informatics, that there are a lot of computational research groups at hospitals and medical schools, at least in the US, that are doing some pretty fundamental work. And so that's what I want to share with you today is really a project that we have been working on for actually quite some time. And so you'll see this in a moment once I start sharing my screen, that, you know, there are some really fundamental computer science and data visualization ideas in this project that are, of course, in that case, also applied to genomics and biomedical data. All right, so I assume you can see my screen. Let me just confirm that. Is that correct?
Yes.
Okay, great. I can no longer see myself in the room, so now I have to trust you. Okay, thank you. All right, so let me get started. This talk is about a grammar that we developed to visualize genome-mapped data. This grammar is called Gosling. And what I should also clarify before I get started what we mean by genome-mapped data. So it's sort of a catch-all term for any kind of data that is visualized in the context of a genomic coordinate system. And this could be literally genome sequences. This could be epigenomic data, so chemical modifications of the DNA in the context of the genomic sequence. It could be gene expression data mapped to the genome. It could be any sort of genomic annotations that tell us about locations of genes or genomic variants and the like. And so we're using this term genome-mapped data because when you just say genomics data, it's a little bit too limiting. But if you're not too well versed in the space of genomics data and molecular biology data, I think genomics is a good stand for genome-mapped data. All right, and what's really important, I think, is to point out that the work that I'll be presenting is really the effort of a very large number of people who were in my lab over the last couple of years. This is not just sort of a one-off project. This is really yeah, as I said, the outcome of many individual projects that are sort of coming together here. And so the team that contributed over the years is shown here. The genome grammar, Gosling, is primarily the work of Sehi Lee, but we're relying on the work of all the other people who are on this slide. And I'll try to sort of point out along the way who did what. Okay, so moving on, I want to motivate this talk by just giving you an idea about the problem space that we're dealing with, and sort of what does genomics data visualization look like in general. And so here is sort of just a very quick overview of all sorts of different kinds of genomics data visualization. And I know these thumbnails are way too small to make out any sort of specific details in these visualizations, but I think it's sufficient to see that genomics data visualization is extremely diverse. There are a very large number of techniques that have been developed over the years to represent data mapped to a genomic coordinate system. And when I say genomic coordinate system, it's really, you can think of the genome as this long sequence, or as in fact as multiple sequences, because every chromosome is its own separate sequence, even though we often don't think about it that way, at least not for data analysis purposes. And then these genomic sequences are annotated with locations of genes and other things. And then we can map data into this coordinate system. So we, for example, know like where a particular sequence belongs within the context of the genome, and that allows us to, for example, understand gene expression data. We know if we find sequencing reads that where they belong in the genome, and then we can quantify this information at the level of genes, or we can find out if there's a genomic variant in a sample that was sequenced, for example. And so here, really, again, the point is genomics data visualization takes many different forms. You probably have seen these sort of circular visualizations, often sort of known as Circos plots, based on the tool that made these really popular. There are genome browsers that have generally sort of a linear representation. And there are many, many other things that you will see in here. And I will try to go into a bit more depth here. But apart from the fact that there are many different ways of visualizing genomics data, there are even more tools that support genomics data visualization. And that makes things a little bit harder, because if you've ever done some data analysis in bioinformatics, or if you've maybe done data analysis in another field, you might be aware that moving data between tools usually generates a lot of overhead in converting data from one format into another, or just even figuring out what is the best tool to actually perform a certain analysis task. And so that's something that, in particular, this conversion issue is sort of a well-known joke in the field of bioinformatics, right? It's about 80% of the time what you're doing is just converting data from one format into another. And that's in fact very true, and something we ideally would like to avoid, right? That's an optimization that we could make if we wouldn't have to convert data all the time. But there are other issues in genomics data visualization that I want to talk about a little bit when we're taking maybe a step back from the tools and just think about visualization in general. So one big issue is that the genome is not organized, at least from a biological point of view, as a linear sequence, right? This is how we represent it often in files or in data. But there are a lot of nonlinear interactions in the genome, right? So there are interactions between, say, a promoter that controls the activation or gene expression for a given gene. There are interactions in some cases between different chromosomes that are either sort of caused by, say, a structural variation in the genome, where one part of a chromosome was inserted into another chromosome in sort of during cell division or some other process. And we're also, of course, these days very much aware that the genome folds in certain specific ways inside of the nucleus, inside the cells. And so this three-dimensional structure also introduces lots of interactions between distant parts of the genome that are generally relevant to understand if you want to understand gene regulation, which is clearly one of the key processes in biology that we're hoping to learn about, because it allows us, for example, to understand disease. It allows us to understand how disease can be treated with drugs by controlling, for example, the expression of particular genes, and so on and so forth. So the main point here is, even though the genome is often represented as a long linear sequence, there are lots of interactions between different parts of the genome, and it's maybe better to think about the genome more like a graph that has a lot of edges with different weights and labels associated with them. Now, another problem when it comes to visualization of genomics data is the fact that we can find patterns in the genome at many different scales. So you could think about one pattern in a genome might be that you're looking at a genome out of a tumor, for example, that's been sequenced, and what you might observe is that a whole chromosome arm, so a fairly large chunk of the genome, has been lost in these cells, so, or a part of the chromosome arm. And that's an example for a pattern that people are interested in seeing. But there are other patterns, again, let's think about cancer, that can be relevant as well, that are a completely different scale. So it could be also a single nucleotide, so a single letter in the DNA of a cell that has changed, that is causing or driving a tumor to grow. And so you can imagine the chromosome arm can be tens of millions of base pairs, while a single nucleotide would be one base pair. And so this is a massive difference in scale. And so that's something that for data visualization is quite a challenge to basically bridge this gap across these scales, and something to think about. And then on top of that, we also have many different types of patterns, right? They could be in the sequence directly. It could be patterns in data mapped to the genome. And I've just listed a couple here. And so there needs to be a certain level of versatility and flexibility in how to represent data so that analysts can find patterns in the data. But this is not the end of it. There are other issues that we need to keep in mind when working with genomics data and looking at genomics data visualization. So in biology, I would say in general, or computational biology, interpretation of a given data set generally relies on bringing in other data sets, annotations to understand what you're looking at. So there's really generally a major need to do data integration, to bring in data from other data sets or data from databases that can tell you more about what you're looking at in a given genomic window, for example. And so as just one basic example here is, you might find that in your data set, a particular gene is expressed at a certain level. So there's some sort of a activity level associated with a given gene. And you might find this in a given condition. Say you find this in let's use the cancer example again, you find that this gene is expressed at a certain level in cancer. In the cancer that you're studying, but you might want to know what is this, how is this gene typically expressed in other tissues, for example, right? So human tissues are very diverse in terms of expression levels. And so you might want to bring in all of this information, and that's often available in public databases. This could be in a collection of data sets for a particular tumor study. And so the more popular your target is, so your gene of interest or a genomic region, the more data you'll find out there that you might want to bring in. And so visualization tools generally need to support this, because sort of contrasting and comparing are really critical tasks in genomics visualization. And I'll say a little bit more about this in a moment. Going on here, another challenge in the genome is that the genome is a very large space. For a human genome, just for context, in case you're not aware of that, it has about 3 billion base pairs, or 3 billion nucleotides. So that's the length of the genome. but for example, only 1% of that sequence is actually encoding proteins, or so it represents genes that then are encoded into proteins. But there are, what that tells you is that at least when we're looking just at genes, the space is extremely sparse, right? So they're really scattered across the genome. And that is something that makes visualization also challenging, because you have long distances essentially between regions of interest in many cases. And so again, it depends a little on what scale you're interested in, but sparsity is definitely something that we are generally struggling with when it comes to genomics visualization, and something to address as well. So finally, there are some more data-related issues. I mean, obviously the data sets are large, although what is large is changing all the time in computer science and when it comes to data. So I think it's still generally an issue, right? Downloading the raw sequencing data for many projects is often impossible. So people would like to do visualization like where the data is, rather than moving the data to their own systems and then looking at the data. So solutions that allow people to visualize the data essentially in situ, if they're sitting somewhere like in an S3 bucket or something, is highly desirable. And finally, of course, with any human data, there are also concerns about privacy, because genomic sequences can be used to identify individuals who maybe did not give permission to be identified as participants, say, in a study. But that's actually a smaller problem for visualization. Now, where does this lead us? So we try to understand this a little bit better. What are people actually trying to do with genomics visualization, and how have they addressed some of these challenges? So what my team did is we developed a number of taxonomies that help us describe genomics visualization. Essentially, going back to this little overview here that I showed you earlier with all these different visualizations, we try to essentially create a way to systematically describe these visualizations. Rather than saying just, oh, this is a Circos plot or is a genome browser, we wanted to find a way to encode this sort of in a more structured way. And so we developed taxonomies, and we did this by reviewing sort of really the literature and several dozens of tools that are out there, and came up with 3 different taxonomies that we use to describe genomics data visualization. One key point I want to make here is that we essentially treated the genome as a coordinate system, and we only considered data visualization approaches that include the genome as a coordinate system. So there is no, we didn't look at tools, for example, like a network visualization tool that would also include maybe genomic sequences like genes and interactions between them, but there is no representation of the sequence. So the sequence and the genomic, the order in which the genome is laid out was a critical piece to all of these tools. And then we came up with 3 different sort of concerns that we wanted to look at individually. One was the data that is being represented. One is the other one is visualization. How is it visually represented? And then the third concern is essentially what kind of tasks, what kind of analysis tasks do these tools actually address? And what kind of analysis tasks do we need to support with genomics data visualization? And you see down here, the data taxonomy is really what data types are out there that people want to visualize in the context of genomics data. The visualization taxonomy, for example, addresses questions like, how is this genome coordinate system laid out? And so the one thing I think that is you've already probably realized is, in a genome browser, we have this linear sequence, and in a Circos plot, we have the circular sequence. Those are 2 different ways of laying out the genomic coordinate system. And then finally, the task taxonomy, as I said, is like what can we accomplish with these tools? What are the questions people solve with them? And so we wrote this up in a paper, and this is sort of one of the key figures from the paper that sort of summarized these taxonomies at a fairly fairly detailed level, although one piece in here, I will mention this in a moment, is not very detailed. In this specific manuscript, or in this paper that we published, and we'll get to that in a moment. So data taxonomy, it seems to be fairly straightforward, right? So we call the data that is mapped to the genome feature sets. And then we have two major types of features. So we can either have sparse features, meaning they are sort of scattered throughout the genome, and we have contiguous features, meaning there is some sort of a measurement for every position in the genome. And then, we can break this up into sort of points or ranges. So a point would be a single nucleotide polymorphism here, or SNP, one position in the genome. For sparse point data, or for example, down here at the bottom, the contiguous range data could be, for example, copy number changes. So that for every position in the genome and every block in the genome, I have a value associated with that. So on top of that, we also have connections between different features in these feature sets. So one clear option is that there is no connection. That's pretty straightforward. And then we might have connections between features. So for example, between genes in a given single genomic coordinate system. So that might be this sort of within connection. This could also be, like as I said earlier, could be contacts of genomic sequences when the genome was folded inside the nucleus in 3 dimensions, right? So there will be connections, essentially edges that give us sort of a strength of the interaction between two locations in the genome. We can have that kind of data. We also have, in some cases, this sort of connections between multiple genomic coordinate systems. So that is something that is relevant, for example, if you're studying like different species, and you want to understand how are our genomes related to each other, what kind of genes can I find in which in genome, and how are they where are they located? So this is the data. The visualization taxonomy is fairly detailed, and I'm going to try to go through this quickly. I don't want to spend too much time on it because I really want to talk about the grammar. But the visualization taxonomy is critical for the grammar because it's really the foundation. So the coordinate system, as I mentioned, there are 4 pieces to that. There's a layout, there's a partition, there's abstraction and arrangement. So the layout, as I mentioned, could be circular, it could be linear. It can also be laid out in a space-filling curve, like a Hilbert curve or a zigzag curve, that essentially where you have a square grid that and you just basically find a curve that visits every cell in this grid at least once or exactly once. So that's another option for a layout of a genome. And finally, there's also, of course, the option to lay out a genome in 3D. If you have some sort of a simulation that allows you to infer the 3D structure that you've maybe measured using a sequencing-based assay, and then you could have a 3D visualization of that. Partition simply describes whether our individual sequences in the genome, so the chromosomes, are sort of connected to each other, or if they're all shown separately. Abstraction refers to the fact that sometimes we look at sequences where we basically look at literally every single nucleotide in a genome. But we can also, for example, when we're looking just at genes, often we're not interested in the parts of a gene that are not encoded into a protein. So all genes are broken up into introns and exons. The introns are the parts that are not converted into protein sequences, not translated into protein sequences, or not even transcribed in many cases. So that is something we might want to remove for visualization purposes, because these introns are often very, very long, and then the exons themselves, so the interesting regions, at least for proteins, are tiny compared to that. So we might want to remove those. And then the third level of abstraction is basically where we scale every part of the sequence to a sort of a standard length. And that could almost be sort of a heat map where we have multiple columns, and each column corresponds to a genomic region, but in the original sequence. So one thing that's really important to point out here is abstraction does not mean that we can reorder any of the sequences. It's really, we maintain the actual order as it exists in the genome. And finally, arrangement is an aspect that that we need to capture in this taxonomy, because we sometimes have multiple representations of a genomic coordinate system. So you could have two genomes in parallel to each other, and this is sort of a scenario where you maybe have a mouse genome and a human genome, and you want to show the synteny between these genomes. So which genes or sequence fragments in human are found in mouse, and where in the mouse genome are they found. So this is essentially what we're seeing here in this between feature set, the connections between multiple sequences. So that can be done, with two sequences in parallel. You could also have them sort of in sequence. You can have a circular layout, and you can have a parallel layout for those, or you can have a circular layout with an arrangement where multiple genomic sequences are in sequence. Or, and this is quite relevant, you can also have sort of two perpendicular genome sequences or genomic coordinate systems. And when you do that, for example, you use the same genomic coordinate system twice, you essentially get a matrix that you can use to show interactions within a single genome. So this is very commonly used to visualize these genomic interaction matrices to understand the 3D structure of the genome. Or you can do it for two different genome sequences. Again, you could compare, say, mouse and human, and then show where the differences are between those genomes and how they're arranged. So this is what's relevant for matrix visualization. So that's just the coordinate system, how that can be laid out. Already that is quite complex in a way, but also the good news is this really describes everything we found in the literature and that was out there in terms of visualizations. And you can basically capture everything if you have these 4 attributes and know their values. Then on top of that, of course, now we need to talk a little bit about how is the data actually encoded. And so the good news here is, and this is the part, by the way, that's not so fully developed in this taxonomy because it's pretty standard fare for anyone who does data visualization. So you have essentially quantitative categorical data that is mapped to some essentially genomic coordinates. And then you can use standard visualization techniques to encode that information. So color, shapes, the y-axis can be used to encode quantities, etc. You can make heat maps and so on and so forth. So this is why we didn't go into much detail here because it's been captured. Then you can also, and this is important, understand, like, so every feature set, so like, for example, the genes is represented as a track in a genomic visualization. That's what we call these things. And so these tracks can either be sort of stacked on top of each other as they are in a genome browser, or you can also overlay tracks on top of each other. And that ability is actually something that is quite powerful, because you can then at that point start combining visual primitives together and make something more complex. I'll show you this in a moment. So this is the alignment of the tracks in the visualization. And finally, something that is really important, although often very much overlooked in genomics data visualization, is how many views are there in your visualization? How are they configured relative to each other? What can these views actually do? Are they like independent of each other, or are they linked somehow? And so obviously, when it comes to views, you can either have a single view. That's sort of a standard thing for a genome browser. In a genome browser, you have one view on your data, and a view in that case defines a genomic region and a certain zoom level, basically. So that's what's shown, and you can only look at that genomic region at a given zoom level, and you have no additional views that would give you maybe a detailed view. But that is possible once you have multiple views, right? Once you have multiple views, you can look at the data at multiple scales. You might have an overview and you have a detailed view. So that's something you can do with two views where the zoom level is independent, but they still would show you the same location in the genome, right? So one shows you maybe a bigger window around the location of interest, and the other one shows you a much smaller window of interest around that location. That sort of changes the scale. Another thing you can do is that you might have multiple views that show you maybe different, or they would show you different locations, or we call this foci in the genome. So you can look at multiple genes, for example, and they might all be shown at the same level of resolution, or you might have the ability to even have different zoom levels for multiple locations in multiple views. So this is also relevant because that is sort of describes sort of more advanced visualizations, allows us to describe more advanced visualizations that have interactive views. I want to essentially skip the task taxonomy because that part I think is quite complex to wrap your head around. It took us a very long time to figure this out. But it is something that I will mention towards the very end of my talk. I just want to make you aware that these tasks are actually really important to understand, you know, what people are actually trying to accomplish with these visualizations. And so this is based on the standard taxonomy of tasks that has been out in the visualization field, at least for quite a while. All right, so I will now move on to tell you more about what you can do once you have sort of found a way to describe a large space of visualizations using taxonomies. Because now we can turn this around, and instead of describing an existing visualization in terms of a taxonomy, we can now create a grammar to go the other way around, right? So we now describe a visualization and generate it rather than looking at a visualization and trying to describe it. And so this is where Gosling comes in. So Gosling is actually an acronym. It stands for Grammar of Scalable Linked Interactive Nucleotide Graphics. And what you see here is just sort of, I think there was a teaser figure from our InfoVis or our Viz paper that just gives you a very, very high level overview of kind of what is possible in terms of visual representations in Gosling. I should also point out, really important here, this is all interactive. So I'm not showing in this figure, but Gosling is really designed to create interactive visualizations, not just static plots that can be included in the paper. And for those of you who are familiar with visualization grammars and familiar with Vega Lite, you will see a lot of similarity between Gosling and those grammars because we've modeled them based on the examples that others in the visualization field have published before us. So this is very, very important to see. And when you look at this example here, sort of the very basic visualization where we essentially have one data set that's just a quantitative measurement along the genome. This is contiguous data and point data, basically, where we have one value per position in the genome. And in genomics, this often comes in form of so-called wiggle files or bigwig files that contain these measurements. And you can see this here at the top. There's a definition of the data. And then we map this data into, we use a mark in this case called a rectangle in this situation. We have the X position defined by attributes from the data file. So position is used to determine the X position of this rectangle. Then the color is determined by a field called peak in that data file. We also define the data type. And then we just define the width of this visualization, so 400 pixels wide and 130 pixels high. And this is the grammar that you need to generate this kind of visualization. Now, really important here, this thing is fully interactive, right? So you could zoom in and out, you can navigate throughout the whole genome in here. And then really quickly, because I'm pretty sure that many of you are familiar with these ideas, so we can easily convert this visualization into a line graph by just changing out the mark that we're using and by changing the peak value. Instead of mapping this into a color for a rectangle, we map it into a y position in this plot, right? So the peak is now mapped to y. And we can keep on going. We can turn this into sort of a points if we want, right? We just change the mark in this case. Or, and this is of course also possible, we can combine these things, right? So now we're adding size and color to these points, and so we have this triple encoding of the data in this case. But that's sort of where we hit a limit, right? So now we have this visualization that looks a bit more advanced than the previous three that I showed to you. But if we wanted to make something more complex, we need to start combining multiple tracks. And so if you remember, I pointed out to you in the taxonomy, right, that we want to be able to align these tracks in different ways. And so this is actually what we do to create more advanced visualizations. And I want to show you this here. So here are two visualizations, two tracks, and we can combine those by overlaying the tracks on top of each other, right? So when we just push this up there, we end up with a visualization like that. You can see the alignment here was added. This is overlay in this case rather than stacked. And then we have two tracks that represent each of these tracks. And you might say, well, you know, couldn't you have implemented this sort of as a special track type? And we probably could have, but we really, in our grammar, we really, and the taxonomy, we wanted to decompose these visualizations or be able to compose visualizations really from primitives, because genomics visualization is extremely rich and diverse. And what people want to do is generally not possible to implement all the specialized track types. And you know, the sort of case in point here is are these genome annotations, like gene structure visualizations. So here, you know, these are just three examples of how different genomic visualization tools represent genes. And these are all created in Gosling by combining multiple tracks and overlaying them on top of each other. So this flexibility is very important, and we can create almost any kind of visualization using this approach. Now, in terms of other representations, right? So here are just a couple of examples I want to go through, and I suspect that if you're in the room in Linz, that these might be a little small. If you're looking at this on your own computer screen, this might work better. This is just really a high-level overview of different visualizations created in Gosling. And in these visualizations are shown in our sort of online editor that you can use to create these visualizations using JSON. That's the language that we use to specify these visualizations. And you see this here on the left. I will not go into much detail. This is something you can actually play around with right after the talk. And see what you find in there. So there are lots and lots of examples, and I'll show you a few. So here's just a standard sort of demo, like what kind of track types can we generate, from heat maps to line bar charts, to stack bar charts and line charts, and so on and so forth. We can also, and this is, you know, really to show the flexibility of Gosling, and you know, you can also create these kind of visualizations. So this is a typical visualization where you have an overview of the genome at the top, and then there are two views below that show you read level information. So individual sequencing rates coming out of a next generation sequencing assay that are mapped to the genome, where you see here at the top, these gray stacked lines represent sort of the coverage, like how many reads are mapping to a given position in the genome, and then you see the individual sequencing rates below. It's kind of hard to see in the blue example, but in the red track, you can actually see there are also differences between the read sequence that's shown here and the actual genome sequence. So this is something that we can all encode using Gosling, and then again, this is fully interactive. You can zoom in and out. You can navigate the whole genome through this. We can create these Circos-style plots as well. In fact, not only we, you can do that too, of course. So I encourage you to check out the examples when I'm done talking. These visualizations here, you know, are 4 different tumor genomes and structural variations and other types of variants, so single nucleotide variants that are shown here. And you can see we can arrange them as multiple views. Each circular representation here is a separate view. These are can be linked to each other. In this case, we created this as a static visualization because it represents a figure from a paper, but they are, it's also possible literally with a single flag to turn a visualization from static to interactive, assuming of course you have the data for the whole genome available. We can create matrices, as I mentioned before, right? So this is an example where we're comparing 2 different assays that were used to infer the structure, the 3D structure of the genome in using 2 different methods, experimental methods. And so here, these 2 matrices are linked to each other. You can navigate them together, and you can see, you know, which method gives you better results in your data. And so this is using the approach that I showed earlier, right, where we have 2 genomic axes that are laid out perpendicular to each other, and then we can map data into those. We can also create these more advanced and somewhat obscure visualization. So this is a visualization technique called Gremlin that was published at a VIS conference like in 2011 by a group at Brown University, and they proposed this as a method to visualize structural variants in genomes. So structural variants are essentially changes in the genome sequence that really affect larger sequence blocks that might be deleted or inverted or translocated from one part of the chromosome into another or from one chromosome into another. And the issue here is, right, so when you look at a genome-wide overview, which is shown at the top here, you might be able to see sort of the locations where these kind of events were happening, but then generally in those cases, you want to zoom in further and investigate individual ones. And they had sort of this proposed 3-level layout where there's like a genome-wide overview at the top, or actually it's a chromosome level overview, so that you see one chromosome, then there's a selected region of interest, and then within that selected region of interest, you can sort of dig deeper and select individual variants and then just see those. And so we can reproduce these kind of visualizations in Gosling. To help you understand a little bit more, how this is structured, I just wanted to talk a little bit more about these tracks and views that I had mentioned. And so views, as I said before, are essentially defining a genomic region that's supposed to be displayed. So it's like basically a start and an end. And essentially a zoom level in that range. And then within a view, you can have multiple tracks, and then by like the tracks inherit basically the location and the zoom level from the view. But you can have different data sets in each track. And as I said, you can overlay them or you can stack them. And then when you have multiple views, you can have different genomic regions and different zoom levels shown in your views. And so this is just an example, how is this visualization structured here? There are, in fact, two combined tracks, essentially. There's the stacked bar chart on top, and then there's this ideogram view below, which is shown in light blue. So it's this thing at the top, and then we have chromosome 2 and chromosome 3 shown below, where the arrangement is a little bit different. So they're in sequence in this case. And so basically, because we can combine these views in different ways, right, we can make them parallel, we can make them in sequence, we can group them in many different ways. We have a lot of flexibility. This is another example for how we overlay tracks, right? So this construct here, which is called an ideogram, that shows a certain regions and chromosomes in different colors. This is based on a very old method where chromosomes were literally dyed using a dye that then showed you sort of the density of certain molecules in the DNA. That's why there's lighter gray and white and black in here. So these are constructed by overlaying 3 different tracks actually on top of each other, right? So there's one that just shows you these bands. There's one for the left triangle, one for the right triangle, and then there's one for the labels. So this is something that we do frequently in Gosling to construct these more advanced visualizations. Here's just another example where we first stack things in the stacked area chart, and then so this is the top track, and then we stack this on top of the tracks in the bottom. So where we have an overlay of the 4 tracks that I just showed you. As I said before, right? So there's this arrangement feature for tracks as well, where we can lay out the genome in circular ways or linear, and then we can combine them in all sorts of ways. So just to illustrate that it's possible and considered in the grammar. One thing that is somewhat unique to Gosling, although it's also been implemented in other tools in the past, is semantic zooming. So here in this little video, you can see when you zoom in these little black lines, as we're zooming, like more structure appears. So this is like, depending on how much space is available, right? These locations, I think these are genes, are shown in a different way. And Gosling supports that as well. So here is sort of the idea, like, when we want to have a genome-wide overview for the genome sequence, we can start stacking, basically the number of, we can create a stacked bar chart for how many A's, C's, G's, and T's, which nucleotides are present in a given region of the genome. And then as we zoom in, we can go all the way down to viewing that information really at the individual nucleotide level. And you can see this here in this movie, right? So as we zoom in, so at some point we have enough space to actually show the individual sequence and even show the nucleotide labels on here. And so this is all implemented by essentially defining breakpoints that you can control in your grammar. So at what zoom level should we use what visual representation? And you basically need to provide these definitions in your grammar. And then you can do things like this, but you can also do things like this example here. Here we're looking at the distribution of a particular type of genomic variants along the genome. We can sort of go from this stack bar chart representation to this lollipop representation, which is very popular and implemented in a tool called ClinVar, or a database, and its video called ClinVar. So this is something that Gosling supports. Something else that is really important to point out is our ability to create multiple link views, right? And so I've talked about this before, how we can have multiple views, and here's sort of an illustration of this. Here are 3 views. There's the circular overview at the top, where we have 2 regions of interest that we can change and scale. And you see there are 2 linked views below that allow us to construct a sort of overview detailed visualization that not only shows us 2 regions, but also then allows, for example, to see the start and the end of these arcs in this visualization. So these are typically structural variants that we want to support. We're also working on responsive visualization design for Gosling. So one problem that you have, the more data you add, you run very quickly out of screen space. And since often it's not clear how much screen space there is, you can define a visualization that will respond to whatever display size there is. And you can see this in this video. It uses the same sort of approach as semantic zooming, but it's based on more of the vertically available screen space rather than the zoom level. Right, and so we can define fairly fancy stuff here. So here's just another example that shows you how this feature can be used. So here are, say, these 2 views. And they are independent of each other in this case, and so we can also come up with things like this, where we then sort of intersperse the views or link them together in this way. Which this doesn't make a lot of sense from a biological point of view, but it illustrates what is possible with Gosling and this responsive design. I want to now spend a brief moment talking about how we've built this, and I'm going to keep this very brief because I think these are some details maybe that are not even that relevant if you're more interested in visualization. But as I said in the beginning, this is really the work of many people. So underlying all of these Gosling visualizations is a system called HiGlass that my team developed a couple of years ago. That was sort of our first attempt at building a genomics visualization tool that is more flexible than the typical genome browsers. And so for HiGlass, we had to develop a system of client-server infrastructure that allows us to access these large genomics data sets very efficiently and to render them in the browser. In order to do this for genomics data, you generally have to pre-process your genomic data in order to essentially allow random access and to create essentially tiled data sets. So very similar to sort of the Slippy map or Google Maps approach, where you have pre-aggregated data sets at different levels of resolution. And so we built this whole infrastructure around HiGlass. And what Gosling actually does, it essentially just builds on top of HiGlass, so we can reuse all of the existing infrastructure. And then we essentially implemented custom tracks in HiGlass that allow us to render these Gosling specifications. So basically, when you write a Gosling specification in JSON, it actually ultimately gets translated into HiGlass specification that's then run in a HiGlass server, but can also be used serverless. So this is very important. So you don't need to run a server necessarily for Gosling. You will need a HiGlass server for certain data types that are just too big to just be loaded into memory in your browser. But it's not a requirement. Going on, Gosling can be used, in our online editor, just sort of if you want to play around with it and create visualization and see what's possible. But it's of course also something that can be embedded into new visualization tools or data analysis tools. So here's just a little example of a tool that we implemented as a demo, how you can integrate a Gosling view here with the React app. Here's another project that we're currently working on. This is a genomics as a tool for structural variant exploration in cancer, where we've developed some fairly advanced Gosling views that are controlled through a number of React components that allow navigation of these data sets. And so what I've shown you so far for Gosling is, there's this JSON spec. Of course, you can write this by hand if you want, but it's not ideal. We have essentially a TypeScript and JavaScript language, a library that we can use to do this programmatically as well if you build your own visualization tool. But many people in the world are never, or most people in fact, are never gonna create a visualization tool for genomics data. Many people who work with genomics data and visualize it do this in the context of data analysis. Say they write some Python code or some R code. And then they need a visualization to look at their outputs. And so this is something we're supporting with as well. And so we have a Python package, Python library called Gauss that essentially allows you to create Gosling-based visualizations in Python on the fly, using everything that Python has to offer. And the approach that we've taken here is very similar to this relationship between Vega Lite and Altair. In fact, we're using the same mechanisms to automatically generate the Gosling Python library based on the Gauss specification. So we don't have to manually update this. We can run some scripts and generate our Python library automatically. And because it integrates with Jupyter Notebooks, it's a very flexible tool to for anyone who does data analysis in Python for genomics data, because they can essentially build on this ecosystem here, right? So in Jupyter or Colab, you would be using this Gauss Python library to generate a Gosling specification, and then the whole same ecosystem of tools is available to you. It's very easy to work with, though. We can easily install this in Google Colab or in Python, a Jupyter Notebook. So you don't really have to worry about this. Looking at this diagram, which looks fairly complex, but we've been able to package this up in a way that it's very reusable. And then, keeping this brief here, what we have here, and this is one important highlight, is the data definitions, right? So when you're doing data analysis on the fly, you often have data structures in memory that you might want to visualize. Like you might have some pandas data frames or something that contains some of your results that you want to map. And so this is something that we focused on. It's not showing in this example, but you can very easily access in-memory data and visualize it in these Gosling visualizations that you construct using Python code. And so here are just some examples. There's a separate website for the Gauss project that has a lot of examples similar to our Gosling editor, and you can play around with it in there. And so you can basically construct the same things that you can do using the JSON spec. But of course, you have the advantage that you can use methods and you can write functions, you can use loops, etc., to construct visualizations much more conveniently than you would do this in JSON. As I said, the data access for local and in-memory was an important addition to Gauss, because it makes this tool much more usable for data analysts who are not thinking about, oh, I have one data set that I now want to share. They're constantly generating new data sets essentially on the fly and want to visualize those. And so we build a little bit of structure into that allows people to use this more effectively. Finally, I want to wrap up because we're only 3 minutes away, I think, from the end of my time here. What are the main takeaways? So because genomics data is so diverse and exists at so many different scales, people generate all these different tools, and we want to be able to support them with Gosling so that people don't have to switch back and forth between many different analytical environments and convert their data. We have a lot of user audiences to support in this space, right? So there are programmers who might use, our JavaScript or JavaScript library or JSON specs to generate visualizations for tools that they build. There are data analysts who might use Python. And, in the future, we also should probably help those people who don't know how to write Python or JavaScript code and, might want to create visualizations through a graphical user interface. What's really important in this is that we need to support, embedding of these tools into the existing data analysis ecosystems, right? So that's why we moved why we built the Python library, because of course nobody does genomics data analysis in JavaScript. So people do this in Python or R, as I said. And we want to give people the tools that fit in with their existing workflows and with their existing toolkits, so that they can use our tools in that context rather than having to convert into another system. Future directions, something I want to point out. So this is a project for genomic recommendations. This is a knowledge-based recommendation system, so it's not any fancy AI, but there's certainly potential for that in the future. And then just to move on, right, there are some other things we could do in the future with this kind of approach, right? So one thing that certainly we can do now is, instead of people building, dozens or hundreds of tools. In fact, there is one list of genomics visualization tools that has about 400 entries or something. We could actually have a platform that allows people to construct whatever visualization they need for a given task, instead of moving back and forth across multiple data sets for every analysis. Because customization is clearly a key feature or key need in genomics data visualization, given the many different data types, the many different questions that people have. And so to make this easier for all audiences, or at least for audiences who don't know how to write code, a graphical user interface to construct visualizations, interactive multi-view visualizations is certainly something that could be helpful there. You could also imagine that now that we know what visualizations look like, we could actually try to train an AI or machine learning model to recognize say a sketch or an image of a genomics visualization and construct the corresponding visualization using Gosling. One challenge that you typically have with these kind of specialized visualizations, like genomics visualizations, right? They are not like as abundant as say bar charts and scatter plots and pie charts. But what we can do with a grammar like Gosling, we can actually generate an enormous training data set that based on the grammar, right? We can just generate all these visualizations, and we can then train image recognition on that, for example. Of course, there are some nuances, some details that I don't want to go into right now, but that's certainly possible, right? We can create that training data set on the fly. And finally, something that I personally am very interested in is, can we build sort of a collaboration core into these, maybe not into the grammar, but maybe into a framework around the grammar that will allow us to construct sort of collaborative visualizations for genome-mapped data, basically with the same ease at which we can, create a Google Docs document together and then work on that collaboratively. So those are just some possible future directions, some of which we're certainly already exploring. And I want to thank you for your time today. I apologize for running over and then for somehow dropping out of the meeting. I hope you'll give this tool and Gosling a try. There's a website that has all resources linked on it. So thank you very much. I am happy to stay here, take questions. I also understand since I'm already ran over time, you might have to go elsewhere.
I'll start with a question. Thanks, Nils, for the talk. Great, super awesome and flexible tool. So I was wondering, one thing that I noticed when we do something with Vega, it's great as long as what you want works. But if what you want doesn't work, you get stuck, right? You get stuck hard. So for example, if you want to add lasso selections into Vega, you have to go through a very complicated process to make sure you're extending the grammar or the backend. Is this a concern in Gosling, or is there a way that we can handle this? Is there maybe a, can I JavaScript anything in addition to that? Is there an interface for that or anything like that?
Yeah, so that's a very good point, Alex. I think this is sort of a question that we have run into this a couple of times, but for us, it meant to extend the, what we can do with Gosling by changing the underlying JavaScript code. There is not really sort of an API for that, so it's not something that anyone could just go in and do. But I think there's also, and I believe this is very important, somewhere I think we have to draw a line. Do we want to turn this into something that can do everything, or is it sufficient if you have something that can do, 90% of what people want to do, which is, is addressing probably the majority of use cases. And so I think we're trying to get to that point. We're trying to find that sweet spot where we know that, the vast majority of people who work with genomics data and need to visualize it can do it. And if there are a couple of people left out in a way, then we need to look into alternatives to, either build a completely custom tool, which we, in our case, can also do because we still have HiGlass behind the scenes. And in HiGlass, you can actually write your own tracks in JavaScript, and you have full flexibility if you want to do something really weird, or if you want to connect like genomics visualization with other types of visualizations. That can always be done using the sort of the components that underlie Gosling. But I think instead of trying to make it extensible, we want to find the sweet spot and make that available to the community.
There's another question from Vaishali. So how does the responsive visualization work?
How does the responsive visualization work? So it's based on the same kind of idea as, like how you build responsive websites. In CSS, you define these breakpoints, and then you transition to a different visual representation at that point. So once your vertical space, for example, is too small, you can define that you want to use a different visual representation. So that's how it's done, very straightforward. The one thing I want to point out here is that, as in a way, a downside of this one thing that's not ideal about the both the semantic zooming and the responsive visualizations is that you often have to implement multiple visualizations, and so these specifications can get very long, and that's because you basically define these different things separately. And so one way how this can be addressed, and it's sort of partially implemented in Gosling right now, are templates. So that certain visualizations are essentially available as a template, and then you can say, okay, once we hit this breakpoint, switch to this template instead of something else, and that way your specifications become a little bit more manageable, because they do. I mean, particularly if you look at the raw JSON, they become incredibly long. The same thing, obviously, also with Vega Lite and Vega, right? So these things are massive. And very hard to understand if you didn't write it yourself. So that's why also we need these kind of tools like the Gauss library for Python to construct these things in a slightly more manageable way.
Yeah, thank you very much. In the interest of time, we have to close the session now, although I would also have a couple of questions. Nils, we can do that offline. So the next speaker will be next month. It's not finalized yet. We will announce that in due time. And let's thank Nils again for a great talk. Thank you, Nils.
Thank you all.
Thanks, Nils.
Thank you.
Bye-bye.
Bye-bye.